For this lab, we're going to focus in on the Kirby Bauer method. The purpose of this test is to perform a culture and antibiotic sensitivity test for a particular organism. For this in lab, we would have you work in pairs. We're going to use a very large plate, and you would inoculate your one plate with one of the following organisms, and you would make a lawn of bacteria over the surface of this plate, and then you would bring it up front to our antibiotic dispensing disc machine to where you would disperse the antibiotics onto the plate, and then you would incubate this plate at 37 degrees Celsius. So what we would normally do is split up a group, and you would get Staph aureus, and so you and your lab partner would be responsible for that. Perhaps your table mates would be responsible for E. coli, and so on and so forth. We are trying to see how each one of these organisms performs to each type of antibiotic presented on the plate.
In this video lesson, we're going to learn how to do the Bauer-Kirby method of antibiotic susceptibility testing. For that method, we use a standard size agar and plate called Mueller-Hinton agar, and these are the large 150 millimeter plates that will hold 12 different antibiotic discs. So the first thing we have to do is inoculate the Mueller-Hinton agar with the organism we're testing. Now in this case, this is one of the bacteria we're using in today's lab, Enterococcus faecalis. So we're going to be using a swab for this. We're going to remove the inoculum by flaming the tube, putting a swab in. Then we're going to wring the swab out against the inside of the tube to get rid of excess liquid. Flame the tube and replace the cap. Now we're ready to streak the plate. Now we divided our plates into thirds. As you'll see in the illustration in your lab manual, to help guide our streaking. And we're going to streak perpendicular to each line, going all the way to the edge of the plate and overlapping the lines, working our way down to the center. So we lift the lid, and we're going to go perpendicular to this line. Notice I'm going all the way to the edge of the plate, and I'm overlapping the lines till I hit the center of the plate. Now we're going to rotate the plate and streak perpendicular to this line. Again, all the way to the edge and all the way to the center. Use the same swab. You don't have to dip it in again. So again, all the way to the edge, overlapping the lines. Till we hit the center. And finally rotate it to the last line and streak perpendicular to that line. All the way to the edge of the plate. And all the way down to the center. And now we've inoculated the plate so that the whole plate will show solid bacterial growth next time. Of course, when we're done with the swabs, they're going to go in the biohazard container. Now we're ready to put on the antibiotic discs. Now, since this is Enterococcus faecalis, a gram-positive bacterium, we're going to use the antibiotic disc dispenser that contains antibiotics, often useful against gram-positive bacteria. You see that the lid is labeled gram-positive. So, very simple to put on the antibiotic discs. We remove the disc dispenser. It also says gram-positive on the handle. And we're going to take the lid off of the plate. Put the antibiotic disc dispenser down over the plate and push down firmly on the handle. And as you see, that dispenses the 12 antibiotic discs, and it also tamps them so they won't fall off when we turn the plate upside down and incubate at 37 degrees Celsius.
So for day 2 of this experiment, we're going to be reading our results. Let's talk about the actual media that we're going to be using for the Kirby Bauer method. So again, the purpose of this is to look at a particular culture and antibiotic sensitivity test for a specific organism, and we tested or will test a number of different organisms. The media that we use for the Kirby Bauer method is the Mueller-Hinton agar. Some things about the Mueller-Hinton agar that are standardized. It has a pH of 7.2 to 7.4. Now, an interesting thing about this is that this is the pH of blood. Why might that be important? Well, we are testing the efficacy of antibiotics, and if antibiotics are going to be used in the human body, we need to see how they're going to be effective at that particular pH. Mueller-Hinton agar is also relatively a soft agar, and that allows for lateral diffusion. So that means that we can have that antibiotic diffusing out through the media and treating the media surrounded. It also is 4 millimeters in depth. That is standardized across the board, again, to allow for that lateral diffusion. Plates are incubated at 37 degrees Celsius. This is human body temperature. Again, if we're testing antibiotics, we want to be able to test them at the temperature in which antibiotics are going to be used, in which organisms are going to be growing. We incubate these plates for 18 hours, so it's less than our usual 48. And that has to do with the fact that a lot of antibiotics are going to be most effective on young organisms or organisms that are early in our growth replication curve, in that lag phase when we are building proteins and building cell walls. So you need young organisms in this antibiotic sensitivity test, and the amount of drug is also controlled. This is responsible for controlling that lateral diffusion of the drug into the media itself, but it also gives us an understanding of what dose of drug is going to be most effective under these controlled circumstances. With the Kirby Bauer method, we are going to be measuring the zone of inhibition, and we're going to be measuring it in millimeters. Now, this type of measurement is going to be different from our disinfectants and antiseptics in the fact that we are going to measure the entire diameter of the clearing. So if you remember, in our disinfectants and antiseptics, we took it from the edge of the disc to the edge of the clearing. Well, that's not the case for this. We're going to take the entire diameter, and so that's all the way across the circle. If you are unable to get the entire diameter measured with your ruler, you'll want to take 2 times the radius. So you'll measure at the middle of your disc out to the edge. That would be your radius, and then you're going to multiply that by 2 because the diameter is 2r. Now with this test, we're going to have 3 different levels of susceptibility. We are going to have drugs that are sensitive. We are going to have drugs that are intermediate, and then we are going to have drugs that are resistant, or organisms that are resistant to the drug. So the definition of sensitive is that a normal dose of that antibiotic is going to be effective. So the dose that is provided on the disc is the dose in which the bacteria is sensitive. Something that's intermediate means that if you're going to use that antibiotic, a higher than normal dose is required. And then something that is resistant means that that antibiotic is no longer effective against that particular species of organism. True or false? All bacteria found within the clear area around the discs are dead. So what that is asking is, just because you see a clear area here, does that mean everything in that clear area is dead? So if you were to culture it, Are any cells viable? The answer to this is false. You could have potentially endospores in there to where, yes, the vegetative cells have died off, but you may have endospores within this zone of inhibition and they survived the drug. They've just gone into that stage of hibernation or survival. Most chemicals don't kill endospores. They're very hard organisms to target. Is take a metric ruler, and we're going to be using this chart and measuring the zones of inhibition on our plate. And the measurement will correspond to the different levels, of susceptibility. So for instance, if we have our disc CF30, that's cefazolin, if you measure a zone of inhibition that is less than or equal to 14, that particular drug or that particular organism is resistant to that particular drug, and so on and so forth. So the next few slides are showing you the results of the test that we would run in lab, and these plates have been oriented in which each disc is in the appropriate area. So for instance, if this is antibiotic disc number 1, this would be number 1 here. If this was 2, this would be 2, and so on, 2, and so on and so forth. Each one of these discs, you'll notice that there's teeny tiny lettering on there. That teeny tiny lettering corresponds to this table here. So that number 1, I could read it, it was NOR10. What you would do is you would take your metric ruler, you would line it up to the edge of the clearing, you'd measure across the entire edge, you'd get a number, and then you would relate that number to the table and see, is that clearing greater than 17 millimeters? Is it between 13 and 16 or 12, less than 12? So as you can see between these two plates, we've got some different responses, different results. And again, we're looking at the difference between two different species of bacteria. So this is E. coli, this is Serratia marcescens. Both of these organisms are gram-negative, but again, they perform differently under different results. You'll also notice that there's some halos here where there's kind of a clearing, but there's not quite a clearing. And so we see that for a number of different antibiotics, but here we see that there's a complete clearing here versus on Serratia, for this one, it's much smaller of a clearing. Here is Pseudomonas and Staphylococcus aureus. Pseudomonas aeruginosa is a gram-negative, Staph is a gram-positive. Pseudomonas, you'll see it's green, and also there are not very many zones of inhibition. This is the only one up here, and we do have some spotty bacterial growth on the edges there, but Pseudomonas is known for being resistant to a number of different antibiotics. Pseudomonas, we talked about this earlier in semester, is a problem in burn patients. They have, they can become infected and then be fighting a Pseudomonas infection. I talked in my lecture about Pseudomonas biofilms on IV catheters and how that was an issue. It's mainly got a lot of resistance factors, and so it's one of those that we keep our eye on. Staph aureus has some interesting things happening. You can notice that Staph aureus, or at least our strain of Staph, has got a zone of inhibition for all of our antibiotics. And then there's something interesting happening down here, which we'll talk about later in the lecture. But Staph, you can see how it responded there. Here's Bacillus meg and Enterococcus faecalis. Both of these are gram-positives, as well as Staph aureus is a gram-positive. But you can see the difference between B. meg and Enterococcus. B. meg, again, these clearings, can we assume that in this zone of inhibition, all of those cells are dead? And the answer is no, because Bacillus megaterium produces endospores. So that is potential. If you were to swab somewhere in here and then try to replate it, you may see that you do end up with growth. And look at the range of the zone of inhibitions. Here's one that's very small. Here's one that's very large. Again, on the practical, we would provide you measurements of that, much like in this photo, where you're able to see, okay, here's 0, and then this looks like 30 millimeters. So this is in centimeters, add a 0, because there are 10 millimeters per centimeter. So the zone of inhibition for this example would be 30 millimeters. Here is our table and our chart. I'm going to fill this in as we go. So if you have your student notes, fill this in. I'm going to fill it in with data from classes past. For cephalotholin, E. coli, we have an intermediate. We have resistant, sensitive, and intermediate resistant. For clindamycin, we have resistant, sensitive, resistant. Erythromycin, we have resistant, sensitive, intermediate. Norfloxacin, we have sensitive, intermediate. Penicillin G, we have resistant, sensitive, and then sensitive intermediate. Sulfa drugs, we have sensitive, resistant, sensitive, resistant. Tetracycline, we have sensitive, resistant, resistant. Trimethoprim, sensitive, resistant, sensitive. Vancomycin, we have resistant, sensitive. I've gone ahead and totaled up our total number of resistant per species of bacteria, and I want to ask some few questions here. Between the different types of organisms, what organisms are we tending to see more susceptible, meaning least resistant, to antibiotics? And that would be our gram-positive, so staph all the way to Enterococcus. These are our gram-positive organisms, and that just has to do with the cell wall and the targets of some of these antibiotics. We have staph and bacillus being more sensitive than some of our gram-negatives. This is an unmutated strain of staph in the lab, so we do have some strains of staph out in the environment that are resistant to a number of different drugs, such as MRSA or MRSA. Enterococcus faecalis is microflora, but we do see some resistance genes in Enterococcus faecalis, and it can be an issue in the hospitals to have an overgrowth of Enterococcus faecalis and then have it be resistant. It's one of the top numbers of infections, death by infections in the hospital. We're seeing Enterococcus strains resistant to vancomycin. For a while there, vancomycin was considered one of the last line antibiotics, and so now we're seeing a normal flora bacteria resistant to vancomycin. Every time we take antibiotics over the course of the lifetime, you have this memory inside of your gut, and so those cells that have been exposed to those antibiotics can have resistance, and so those resistance genes can get spread. But on a whole, our gram-positives are generally more susceptible to the antibiotics that we tested versus our gram-negatives. We have more resistant strains. And then Pseudomonas being our number one resistant strain. Overall, we have 19 resistant gram-negatives resistant drugs, and 9 that are resistant in our gram-positives. I've highlighted a column here in blue, and there's a question that I'd like to ask you, and there's no right or wrong answer to this question. It's just more of an ethical question, more of one of those let's think about it question. For Enterococcus faecalis, if you were a prescribing practitioner, would you prescribe penicillin G? Now, I've had students argue both sides of this, and some students argue, yes, prescribe it because it does work, just prescribe it at a higher dose because it does tend to be on that borderline intermediate, or some strains are showing intermediate. Another argument is, yeah, prescribe it because we're seeing a lot of intermediate and resistant strains to these drugs. So yes, you have to use what works. An argument against, I've had students say, well, if it's showing intermediate, then we should use a drug that, is more active, that is the organism's more sensitive to that particular drug. Again, there's no right or wrong answer. There are drugs that are used for specific infections and things like that. There are prescribing recommendations, but it's something that you have to run into, especially when you get into resistant strains. What's going to be the next drug of choice? The other question I have is, what strain is most susceptible and what strain is most resistant? And so one of our most susceptible strains is going to be Staph aureus, and that again has to do with that cell wall. Pseudomonas aeruginosa has just got a lot of resistance genes on it. This figure is in the Tortora book, but it's just showing you the difference in cell structure or overall microorganism structure as to what makes an organism most resistant to least resistant. So prions or prions, however you want to say that, are those proteinaceous infectious particles. So they're essentially a misformed protein, and they're a cause of a lot of neurological diseases. Bovine spongiform encephalopathy, you may know it as mad cow disease, Crutchfield-Jacobs disease. Prions or prions are an infectious, kind of a majorly scary infection. In order to destroy prions, you have to treat that particular equipment with proteases, which are going to be enzymes that digest proteins, as well as expose the particular, again, materials, supplies, to a higher autoclave temperature for a longer period of time. Endospores are the environmentally hardy version of these organisms, and they have that keratin protein that protects them from a lot of chemicals, as well as dehydration and things like that. Mycobacterium has that waxy outer mycolic acid, which helps protect them. Cysts of protozoa. Cysts are the environmentally resistant resting stage of a protozoa. So they have a protective outer coat, which is their thick cell wall that's going to allow them to survive longer in the environment. Gram-negative bacteria have an outer membrane, which is super protective for those types of cells. As we get into viruses without envelopes or our naked viruses, they're just environmentally more hardy. Gram-positive bacteria and viruses with lipid envelopes are generally not resistant. They're pretty sensitive to drugs and disinfectants, antiseptics, if we're talking about expanding this to our previous lab. Some definitions that you need to know and be able to interpret to our results. A broad spectrum antibiotic versus a narrow spectrum antibiotic. So a broad spectrum antibiotic is going to kill or inhibit the growth of many different species. So let's say both gram-positives and gram-negatives. It's going to be a drug that is used most favorably because it's going to target a lot of things all at once. Narrow spectrum drugs are going to kill or inhibit very specific species of bacteria, so perhaps only gram-positives, or perhaps gram-positives, but it really only targets a specific type of cell in gram-positives. The narrow spectrums are going to be used, I would say, less frequently because you really need to target in what type of infection, versus a broad spectrum you may see used a little bit more often because it's going to target a number of different types of cells. So when we're looking back at our results, the difference between broad spectrum and narrow spectrum, well, we really need to take a look at our table and say, where did we see across in the rows, not the columns, but the rows, what is a drug on here that was targeting both gram-positives and gram-negatives, and across the board we see relative sensitivity. Norfloxacin is a drug that we would be considering broad spectrum because, again, it's sensitive, and intermediate, meaning you just have to use it at a higher dose. So that would be considered a broad spectrum. A narrow spectrum perhaps would be, according to our data, clindamycin, very narrow spectrum, where we only see it effective against Staph aureus. On a clinical perspective, we use clindamycin in the veterinary medicine a lot of times for bone infections because it's one of the antibiotics that can get deep down into the bone. Clindamycin is commonly used as a topical antibiotic for acne and things like that. So let's get into this idea of synergy, and we saw this on one of our plates. So drug synergy is when drugs work together better than they would on their own. And I like to represent this as numbers. So if we have a drug that is effective, let's say at 4 by itself, and the other drug is effective at 3 by itself, when you use them combined, so using them as a drug cocktail or being able to combine them, the effectiveness is even more. So it would be a 10. Now I know 4 plus 3 is 7, but drug synergy means it works better than that. So it would be an effectiveness of 10. And again, those are arbitrary numbers, but it just kind of goes to show that when you use them, they're more effective together. We are seeing drug synergy on this picture here with 2 different antibiotic discs, and the drug synergy is being displayed up here and down here. So it's showing you in these little dotted lines how the drug performs on its own, and then this big wide area is that synergy. It's working better together. For our Staph aureus, we have 2 drugs on here that are synergistic, and that is this area right down here. And this drug right here is TNP, or trimethoprim, and this is penicillin G. Both of these drugs, or excuse me, not penicillin G, this is our sulfa drug. Sulfa. Both of these drugs are used in combination a lot of times because they target the same metabolic pathway. So the metabolic pathway that it's going to target is the synthesis of folic acid. Now for us as humans, we get folic acid from our diet. For bacteria, they have to make it, and it's essential for them. So they have to make it. If they can't make it, they can't make DNA and RNA. So this is a metabolic pathway controlled by enzymes with a lot of intermediates, but the essential nutrient is PABA, or PABA. That stands for para-aminobenzoic acid, PABA. That's the nutrient, and it's used by a number of different bacteria to synthesize folic acid. So I want us to focus in on the yellow before we focus in on the orange. So the normal metabolic pathway would be PABA going to dihydrofolic acid, going to tetrahydrofolic acid, and then tetrahydrofolic acid would eventually go on to make DNA or RNA. Now, we have two drugs, trimethoprim and sulfa drugs, that, again, a lot of times are used in combination. Sulfa drugs are going to act as a competitive inhibitor. So refer back to our chapter on metabolism on competitive inhibitors. Sulfa looks very much like our PABA, and it competes for the active site of the enzyme that will produce dihydrofolic acid. Trimethoprim interacts with dihydrofolic acid turning into tetrahydrofolic acid. How? It too is a competitive inhibitor. So it acts as the substrate to our active site. So both sulfa and trimethoprim taken together use less than what they would need on their own. So Bactrim, maybe you have heard of that drug, is a drug where sulfa and trimethoprim are combined, and so you actually need less of the drug dosed in order to get an effect because of that drug synergism. So we see that on our plate here, because let me outline in blue. Here's how our organism would perform normally, or a drug would perform normally, but again, we see this synergistic effect happening in combination.